Today we're going to learn about mRNA translation, one of the necessary steps of metabolic functioning that comes after the process of mRNA transcription. Without transcription and translation, your body would have no possible way to make proteins or function. Proteins allow your body to do everything.
First, DNA must be transcribed into mRNA, which is shown here. Then it has to be translated into amino acids through a three-step process in order to create a polypeptide chain, aka a protein. The first step of translation is known as initiation. To begin, two ribosomes attach to the mRNA, allowing for the first tRNA to bind to it at the start codon, AUG, at the P site, which signals the beginning of translation. Once translation is initiated, the next step of elongation can begin. tRNA is able to attach the codons of the mRNA using anticodons. Anticodons have nucleotides that are opposite base pairings to the mRNA codons, which allows for the correct amino acid to be added to the chain. In our model, A is purple, U is blue, C is yellow, and G is green. Remember that since this is mRNA, we don't have any T's. If we know that A pairs with U and C pairs with G, then we can figure out the anticodons. For example, the anticodon for AUG is UAC. Now the second tRNA can attach to the A site. This is the beginning of the formation of an amino acid chain. Notice that the first tRNA has shifted to the E site and the second tRNA has shifted to the P site.
Then, the third tRNA enters the ribosome and attaches to the next codon in the A site after the mRNA has moved.
Notice that the second tRNA's amino acid is being added to the polypeptide chain. And now, the first tRNA has exited the ribosome. This shifting of tRNA and attaching amino acids at the P site continues. The process repeats until the amino acid chain is fully formed.
Just a reminder that the amino acids, which are represented by the beads, would not be a part of the tRNA after the amino acid has attached to the polypeptide chain.
Okay, now that you have thrown your tRNA, the polypeptide chain is almost finished. The last step of translation, known as termination, can occur. In termination, a tRNA has attached to the stop codon, in this case UAA, of the mRNA, signifying that the polypeptide is ready to be finished and it can get folded into a protein. This final shifting occurs and the tRNA exits the ribosome, and the last amino acid is added to the chain. Finally, the very last tRNA exits the ribosome and the amino acid chain is finished. Since the mRNA isn't needed anymore, it also exits the ribosome.
Keep in mind that this is a model, so in our body, these processes are happening on a minuscule scale and way more amino acids are needed to make a protein. Don't forget to say a quick thank you to your cells for protecting your DNA and coding for the amino acids that keep you alive. Thanks for watching!
Hi! Oh my god, you should probably take the stuff on the floor.